Barbara McClintock almost didn't go to college. It was 1919, and her mother, deeply rooted in the societal norms of the time, believed that obtaining a college degree would harm her chances of securing a good marriage. At the time, the role of women was largely confined to the domestic sphere, and higher education was not seen as a priority for women. Yet, Barbara's father, returning from military service, stepped in, advocating for her education. His belief in her potential was unwavering, and it would prove to be a defining moment in the history of science. With her father's support, Barbara enrolled in Cornell University, a decision that would change not only the course of her life, but the future of genetics. She enrolled at Cornell University's College of Agriculture, where she quickly became captivated by genetics, a new and little understood field. It was here that she developed an insatiable curiosity about the chromosomes of maize, also known as corn. As Barbara once said, no two plants are exactly alike. They're all different, and as a consequence, you have to know that difference. Many characteristics of organisms are determined by heredity, their genes, which are stored in the chromosomes inside their cells' nuclei. Barbara McClintock studied corn's hereditary characteristics, such as the different colors of its kernels. She explored how these traits were passed down through generations and linked them to the changes in the plant's chromosomes. Her discovery of transposable elements in Zea mays completely changed the way scientists think about genetic patterns of inheritance. McClintock's observation of the behavior of kernel color alleles was revolutionary in its proposition that genomic replication does not always follow a consistent pattern. Subsequently, it is, as a result of both autonomous and activator-controlled transposition at different stages of seed development, that the genes of maize kernels are capable of producing a variety of color patterns. In the 1940s and 50s, McClintock made a groundbreaking discovery. Genetic elements could sometimes change position on a chromosome, causing nearby genes to become active or inactive. This revolutionary concept, known as transposition, would ultimately reshape our understanding of genetics and revolutionary biology. McClintock's passion led her to earn her bachelor's, master's, and doctoral degrees at Cornell. Her scientific journey took shape in 1921 when she attended the undergraduate genetics course offered at Cornell, taught by C.V. Hutchington. Genetics was still an emerging field, and many biologists were hesitant to accept its revolutionary, but out there for the times, concepts. Hutchington, who recognized McClintock's exceptional interest, invited her to participate in a graduate-level genetics course. This pivotal moment cemented her path in genetics and kick-started a revolutionary change in the field. At the same time, she was studying cytology with Lester W. Sharp, who focused on chromosome structures and their behavior during cell division. Chromosomes fascinated McClintock, as they are known to carry hereditary factors. By the time she graduated, her direction was clear. She decided that she would devote her life to cytogenetics, the study of chromosomes and gene expression, a fitting mix between her two major influences. After completing her PhD in 1927, McClintock remained at Cornell, working with Charles R. Burnham, a plant cytogeneticist, to link each of maize's 10 chromosomes with the genes they carried. During this time, she formed close scientific bond with future Nobel laureate George Beadle and a geneticist, Markle Rhodes. Their collaboration created an academically rich environment that shaped her groundbreaking research. Although McClintock loved her research, she disliked the distractions of teaching. In 1941, she left teaching for Cold Spring Harbor Laboratory, a place where she could fully dedicate herself to scientific discovery. It was here that McClintock made her most astonishing discovery. She observed patterns in maize that didn't follow traditional genetic rules. She found that genes weren't fixed in place. They could actually jump from one location to another, turning surrounding traits on and off. This was genetic transposition, a completely revolutionary concept. But the scientific community wasn't ready. When she presented her findings in 1951, her colleagues were dismissive, even skeptical. They thought I was crazy, she later recalled. I just knew I was right. She stopped trying to convince others. Instead, she focused on what she loved, her research. For over a decade, she worked in relative isolation, trusting the evidence under her microscope and not letting anyone's opinion affect her research. Her patience and confidence paid off. In the 1960s, a new research in molecular biology finally proved her right. The world had finally caught up to Barbara McClintock. Her discovery of transposable elements, jumping genes, transformed our understanding of genetics, influencing fields from evolutionary biology to medical research. Today, transposons are recognized as essential components of DNA that affect gene expression, adaptation, and even disease. Some modern research studies are being done on transposable elements containing testing of RNA transposons, which function via reverse transcription of an RNA intermediate. This class is recently seen to have a huge impact on mammalian genomes and could have potential use as mutagens. It was in 1983 that McClintock was awarded the Nobel Prize in Physiology or Medicine, which was more than 30 years after her groundbreaking discovery. She was the first woman to win the award unshared in that category.
Prize in Physiology or Medicine from His Majesty the King.
But for McClintock, the recognition wasn't the reward. The reward was the work itself. Beyond her discoveries, her legacy was one that highlights persistence. Her ability to trust her own observations, even when dismissed by her peers, is a lesson for scientists everywhere. As McClintock once said, "If you know you're on the right track, nobody can turn you off." Barbara McClintock spent her life not just uncovering the secrets of genetics, but demonstrating the power of scientific curiosity, resilience, and an unwavering commitment to the truth that science holds.